Oh, okay, thank you. Sorry about for that little delay. So last week we spoke, we had a kind of introduction to the project, right? And I gave you guys some assignments. I'm sure, I think I got like two or three people. So if you haven't submitted the assignment, please, I give you the grace before the end of today to please send that to me. Before the end of today, kindly send that to me. That's one. So do we is who was our captain, our class rep yesterday? Is it Binaj or Umar? Who was it? Who was the class? Who was this group captain? Was it Umar or Binaj? I can't remember. Can you hear me?
I don't
I believe
believe.
it was Deja.
Okay, it was Umar, right? Hello, I'm not hearing your guys' voices. You're not speaking.
From my understanding, I thought it was Deja, or I could be wrong, but I felt like it was Deja maybe.
Was it
Oh, okay.
Aaliyah?
It was Aliyah, right?
Yeah, for the class
Yeah, I
leader,
think so.
right?
Okay, yeah, I think it was Aliyah.
Yeah.
Okay. Because I want her to be taken register for every of our meeting. Why we are saying doing that is our sponsor want us to have record of that, right? So that's why I want us to do that. Okay, let's continue. So we had an introduction to the class last week. So and I said this week we are going to be looking at protein structures, right? Because we made it clear to us that interactions between an organism and the host, for instance, between SARS-CoV-2 and human, has to do with proteins interacting with each other. And I give an example that a particular protein on SARS-CoV-2, which is called the spike protein, will interact with a protein on the human cell, which is the angiotensin converting enzymes. So the two of them comes together, right? They interact, forming a kind of bond, right? And that gives access to SARS-CoV-2 to be able to get access into the human body, correct? And be able to produce and replicate itself. So, and I observed that we need to go a little bit into structure of protein, right? And the last time I showed you a 3D structure of a protein, right? I think for SARS-CoV-2 spike. So today we are going to look at proteins, amino acid, and structures, right? Now, this is a typical, and I gave you that assignment, and I'm sure if you have done it, you should have seen this structure that I'm showing to you. This is the structure of this is every amino acid has three main functional groups, right? Number one is the amine group, NH2, right? Number two is the carboxylic group, COOH, right? And the number three is the side chain. What did I call it? The side chain. So if you look at the assignment I give to you, you would have observed something that all your amino acid has this amine group, acid group, this hydrogen group, but the side chain differs. Is that correct? So all amino acids are the same. They have the same amino group, carboxylic group, hydrogen group, but the side chain is what differentiates them from one another. And let me give you the first one that we have here is glycine. Glycine is the simplest of all amino acids, right? It has an amide group, right? A carboxylic group, hydrogen, the regular hydrogen atom. Then on the side chain, it has hydrogen atom. That is the simplest of all amino acids, glycine. And the three-letter code for glycine is GLY. And the single letter code is G, right? Now, another example is aspartic acid. It's another essential 20 amino acid, one of them. And if you look at this also, you will see the regular amine group, carboxylic group, the hydrogen. But if you look at the side chain, the side chain here for aspartic acid differs from that of glycine, right? So all amino acid has carboxylic group, they have amine group, the hydrogen atom, and they have their respective side chain, right? Let me give one another example that I have here, which is phenylalanine, right? Phenylalanine, as usual, has amine group, carboxylic group, hydrogen atom, and can you see the benzene ring with CHCH as the side chain? So all of them differs by their what? By their side chain, right? So, and why are we looking at this is, if you are looking at protein, protein is a big structure, right? But that protein structure we're talking about is built by amino acid. So when you have many amino acids coming together, right, you have a big protein. Now, that's one. Another thing is the structure of the protein is going to be dependent on the number of amino acid that comes together and the type of amino acid that binds together to form different kind of reaction. Let me give an example. This is a protein, right? And that was the question, I think it was Alia. I don't know, remember the person precisely last time, that why do we have the ribbon? And I said, we are going to talk about the structure of amino acid, right? So please, if you have any question, always let me know. Right. So we spoke about proteins last week. Proteins are large molecules, right? They are made up of amino acid chains, correct? And that's why I gave you what amino acid looks like. And I gave an assignment last time that you should know, give me the 20 amino acids, their 3-letter code, and their 1-letter code, right? So they are found in every cell in the body. They are in every cell of the body. Proteins are there, right? Even in plants, even in microorganisms, right? And they are involved in most of the body's functions and life process. You are able to walk because a particular protein is working. You are able to see. You are able to eat your food and digest because certain proteins, either in form of a macromolecule or in form of an enzyme, right, are performing well, right? And the sequence of amino acid is determined by DNA. Right. So the sequence of an amino acid is determined by what? By DNA. So the DNA that you carry in your body has the information on what type of amino acid should make the amylase enzyme in your mouth that metabolize carbohydrates. So the DNA in our body code for specific protein. So the DNA carries the information for each amino acid. Now, and I've tried to now structure of proteins now. The proteins are made up of amino acids, like I said earlier, right? So when you have one amino acid joined to another amino acid, to another amino acid, you are building a protein, right? And these are joined together by what is called a peptide bond. So I'm going to show you what a peptide bond means. So we have peptides fewer than any that is when the amino acid that comes together to form a protein is less than 50. We refer to them as peptides. So when you have 10, 40, 50, that's peptides. So maybe alanine binds to glycine, binds to arginine, valine, as many, right? And if they are less than 50, that is a peptide. So when you have 2 amino acids, maybe glycine binds to alanine, that is dipeptide. That's 2 amino acids. Is that correct? Dipeptide. Do we have the charts? Yes, I'm going to provide the slides and the recording. Yes, I will provide you guys with the slide and the recording. Thank you for asking. So when you have 2 amino acids coming together, it's called dipeptide. When you have 3 amino acids, it's called tripeptide. Just di means 2, tri means 3. When you have more than 10, we have poly. Poly means many, polypeptide. So when we can now say this is a protein, is when you now have more than 50 amino acids coming together, then you now have a big protein structure, right? So typically, a protein can be from 100 to 10,000 amino acids. Yes, in our body, between 100 to 10,000 amino acids linked together, right? And I've said to you earlier that DNA is the one that carries information for each amino acid. Do you know why? every protein, every enzymes or every protein. For instance, let me give an example. Structure the protein sequence of hemoglobin. How many of us know hemoglobin? Hemoglobin is what our body is present in our blood that carries oxygen, right? So there are certain amino acids. So the amino acid have sequence. So let's say it is glycine, adenine, alanine. There shouldn't be a mismatch. They must always be glycine, adenine, alanine. Glycine, adenine, alanine. I'm just giving an example. If there is glycine and there is another one apart from alanine, there's going to be a problem. And there has been a lot of medical disease that people have now just because there is a misarrangement of maybe one amino acid. Maybe instead of it to have alanine, it has arginine. There is a problem. So so for hemoglobin, hemoglobin has its own sequence, right? So there is a DNA in our body that has the information on how to produce hemoglobin protein, right? There's a different DNA that carries the information on how to produce maybe insulin, right? So DNA carries the information to code for every protein. Are you getting me? So let me now give you an example. In the case of people with sickle cell, people with sickle cell had a misplacement of a particular amino acid. I think it's glutamate. I can't remember specifically, but a particular amino acid, I think glutamine replaced valine. I can't remember precisely. That's why those people have what is called sickle cell disease. So when there is a misarrangement in that thing, there's going to be problem and it can lead to a severe medical condition. So DNA carries the information on what number and the sequence of amino acid that is needed to produce to form a particular protein, right? And I've said that amino acids are composed of carbon. You can see carbon in that structure, hydrogen, oxygen, nitrogen. And in some cases, like if you look, remember the structure I asked you to look at, if you look at cysteine, methionine, they also have sulfur in addition to carbon, hydrogen, oxygen, and nitrogen, right? Any questions so far? Do you have any question? Right. So I've shown you this before. This is the anatomy of an amino acid. It has an amine group, carboxylic group, hydrogen atom, and the side chain. And I said the difference in all of them is the side chain. Okay. So now, how I was telling you that one amino acid can bind to another amino acid. Yes. And what that means is the carboxylic group, right? So just 1 second. I think my let me pause the recording. I think my laptop. Let me get. So just give me 1 second. All right, sorry, I'm back. So okay, let me resume. So how do one amino acid form bond with another amino acid? And that is simple. The carboxylic group, that's the COOH, on one amino acid will form what is called a peptide bond with an amine group of another amino acid. And in that formation, there's going to be what is called condensation. That means there's going to be release of water. There's going to be what? Release of water. So let me show you. Now, this is an example. This is an amino acid. Can you see this? So this is the amine group, right? This is glycine, right? This is the carboxylic group. Now, this is another amino acid, and this is phenylalanine. Are you getting me? Now, when this hydroxyl group, when the carboxylic group here interact with amine group on phenylalanine, they come together. This OH from this group an hydrogen from amine group will be released, right? That's water condensation. So when they are released, now you can now link this together with this, and that is what you see here. Can you see that? That is how one amino acid binds to another amino acid. So when there is going to be there will be a release of water. So, and that for you to get that water, hydroxyl group OH is coming from the carboxylic end of one amino acid, then hydrogen atom is coming from amine group of another amino acid to release what? Water. Then the two of them can bind. So just imagine in your mind and take out these two, right? Do you get that? Take out these two, then this and this are binding. That's what you see there. That is what is called a peptide bond. So when one amino acid bond with another amino acid to make a bond, thereby releasing water, that is a peptide bond. So a peptide bond formed by condensation when the acid group, carboxylate and amine group of two different amino acids join and release a molecule of water. And that is what you have seen here. That is a peptide bond. Now, how do you break protein too? How do you break this bond? The best way to break it is what? Is through hydrolysis. So when peptide bonds are broken by hydrolysis, the hydroxyl group and hydrogen group from water are added. So when you join them together, you take water out of it. If you want to break them, you add water to it, right? Any questions so far before we proceed? Any question? Any question on anything that is not clear about structure of amino acid and how it forms peptide bond to produce peptides or to produce multiple chain of amino acid. Any question? Okay. Now, these are the 20 amino acid, right? That I asked you to write: histidine, HIS, isoleucine, leucine. Also, these are the 20 amino acid. So some people categorize them as essential and non-essential. And what they mean by that is the essential are usually the one that you get from your food. Yes. While the non-essential are the one that the body can possibly produce. Are you getting me? So for you to do protein structure, MD simulation, a 3D structure, you have to know these amino acids, know their names, and at least know their 3-letter words. Now, so we have spoken about protein generally. We've spoken about amino acids and how they come together to form a peptide chain, right? And we have spoken about the functional group that you can see on a typical amino acid. Now, to answer the question that Omar asked last time, that why do we have that big ribbon structure? Now let's now go about different structure of proteins. There is what is called a primary structure, there is secondary structure, there is tertiary structure, and there is quaternary structure, right? So each of them has to do with level of complexity, right? So when you have one amino acid to another amino acid, the complex of the structure is simple, right? So let's go. So the features and functions of protein structures, right? So we have the primary sequence of amino acid. We have the secondary interactions from form coils and sheets. So don't let me dwell much. Now, another thing you need to understand is this. When proteins are forming together, when you have carboxylic group binding with amino group of another amino acid, they form a peptide bond. However, there is tendency for other groups on that proteins, when it is getting bigger, to form a kind of interactions with each other. Are you getting me? So let me give you a typical example. So let's say we have alanine binding with, like the example we gave. Let me go back to this example. Sorry. Now, look at this now. This is glycine, right? Forming a peptide bond with phenylalanine, right? The peptide bond is this one. Now, if this chain continue, right, and it begins to coil, there is probability that this particular benzene, a functional group on this benzene ring, right, will interact, not forming a bond, but interact with this amine, this particular amine group, right? And when they do that, it dictates the structure of the protein. Are you getting me? It dictates the structure of the protein because the more the sequence is bigger, the more the molecule is trying to give a shape, right? And the more it's going to give a shape, the more all those side chains are able to interact with themselves, right? So let me go back. So part of these interactions can be ionic interactions, and this happens between oppositely charged residues. When anytime you see me using residues, I'm just talking about amino acids in the literary sense, right? So for instance, maybe there's a positively charged atom of one amino acid now forming a kind of electrostatic interaction with a negatively charged from another amino acid. There is also hydrogenic charges, hydrogen bonds. They are weaker bonds and they are partially positive charge. There are van der Waal hydrophobic residues interactions, right? I will show you what I mean, this example of this by the time we are looking at the protein structures under chimera. Now, let me now go back to what I was talking about. Now, let's look at this. Let's look at this. This is a sequence of amino acid, right? Are you following me? So let's assume this is alanine, glycine, isoleucine, asparagine, and methionine, lysine, valine. All those 20 amino acids were arranged in this form, right? Now, as the chain is, can you see? This is the first one, right? As the chain is getting longer, the side chain on this particular amino acid is now attracting each other with another side chain on this amino acid. Did you get that now? Are you guys following? Hello? So when the complex is getting bigger, when the sequence is getting bigger, there is tendency, in fact, not there is higher probability that the side chain from one amino acid will find another amino acid that has an attracting interaction. So by the time that is happening, it will be forming the structure of the protein. So look at this. This is supposed to be a straight chain, right? But because this side chain on this amino acid is forming a kind of interaction or bond with a side chain on this amino acid, can you see how it forces this chain to bend? Can you see that? It forces that chain to do what? To bend. Now, if you look at this place, so if in this case now, this is a typical hydrogen bond. If you look at this place, there is an hydrophobic interactions, right, between the side chain on this amino acid with another side chain on this amino acid, thereby forming a curve. So the ribbon I showed to you last time, right? I think I'm going to show that now. If the ribbon is forming because as the sequence is getting bigger, all those proteins side chains on the amino acid were forming interactions with each other, right? This is another kind of bond called disulfide bond. Disulfide, that's two sulfur. Disulfide, sulfide means sulfur. Two. So the sulfur group, and this is likely to be between either methionine, histidine, and cysteine, right? So when this sulfide group on one amino acid is forming another sulfur group interaction with another amino acid, can you see how the two of them come together? And that's how what was supposed to be a long chain begin to interact together, and you see the protein coiling. The protein what? Coiling. So when you now when you have, now let me give you an example. So look at this, look at the primary structure. Primary structure has a simple coiling, a simple interactions between side chain of one amino acid and another amino acid, right? And you can see it's just this curve is formed because probably a functional group on valine is interacting with another side chain on leucine. Now, as the sequence is getting bigger, right, and there is more tendency for more interaction, can you see how this form what is called an helix, like a coil? A what? A coil. So because more side chain of this amino acid are now interacting more with each other, that is what is called a coil. Now, that's secondary. Now look at tertiary. The more, so imagine the more you are having a more coil, that more coil will begin to give you like you can have what is called a loop, a complex structure. So we expect secondary structure to have more complex than primary, tertiary to have more complex than secondary, and quaternary. So when you now have many, so let me put an example. When you have this single primary, that's primary. When you have 2, 3 primary, you have secondary, right? When you have 2, 3 secondary, you have tertiary, right? When you now have 2, 3 tertiary, so this particular one is just this one. So when you now have 3, 4, 1, 2, 3, many of tertiary, that's when you have quaternary structure, right? And these things are happening because of more amino acids and more interactions between the side chain of those amino acids, of one amino acid with another amino acid, right? Any questions so far? Come out. Any question? Either on the chat, you can put it on the chat. So do I want to believe now that we understand? I want to believe now that we understand the structure of protein, right now. Any opinion, any question? Please ask me question if you have question. Yes. Please feel free to ask me question if you have any question. So let me now give you an overview now. So proteins are chains of combination of amino, because if you do not understand protein structure, right, by the time we start working on structures, using some AI tools, learning programming language to applying them to protein, right? You will understand what you are doing. So that's why it took time to just have this particular class, basically for protein structure. So proteins are chains of combination of many amino acids, right? And amino acids are made up of carbon, hydrogen, oxygen, nitrogen, and sometimes sulfur. So if you look at the assignment I gave to you, if you look at histidine, methionine, and cysteine, you will see those are the ones that have sulfur, right? In addition to the hydrogen, oxygen, nitrogen, and carbon. Now, unique amino acid consists of carbon, we have said, carbon with carboxylic group, hydrogen, nitrogen containing amide group, and a unique side chain. And I said there are 20 side chain for 20 unique amino acids. Amino acids link together with peptide bond by condensation, and you can break them also by hydrolysis. So it is possible for protein to be destroyed, right? Yes, that protein can be destroyed. And that is why if you have meat at home and maybe you forgot to put the meat in the fridge, right? And after sometimes you later find out, oh, this meat is not in the fridge. And you eventually look at it and you observe that, oh, it's already smelly. Do you know what is happening? Because the protein on that meat are being broken down by organism, right? So protein can be denatured either by chemicals, either by heat. Yes, excessive heat can destroy protein structure, right? Chemical, heat, and some other factors, right, can destroy protein structure. Any question on protein structure? So I've explained to you. So let me show you now. Okay, I even have it here. So Another quick review I also have here is attraction and interaction between the side chain cause the protein to fold. Yes, I've said it to you before. So a kind of attraction and interaction between the side chain causes the protein to fold. So if there are more attraction and interaction, if there are less, you will have primary structure. If there are more, you will see primary structure forming helix, which is secondary. If there are more, you will see secondary forming tertiary. If there are more, you will see tertiary forming what? Forming quaternary structure, right? So that's how the structure are being determined. And protein shape determines its function. Yes. Another thing I want you to know here, the shape of a protein will determine the function of that protein. Oh, let me say it again. The sequence, number one, the sequence of the protein, the number of the amino acid will determine the shape of a protein. Therefore, the shape of a protein will determine its function. Oh, please, take note of that. Take note of that, you need that a lot, right? You need that a lot. Again, let me say it. The amino acid type and the number will determine the shape of a protein. And the shape of a protein determines the function of the protein. So that is exactly. So protein can be denatured or destroyed or have their shape changed by heat, acid, I said chemical. So heat, chemical like acid, base, or salt, and mechanical agitation. When there is a serious, rigorous mechanical agitation, it can destroy the shape and the nature of a protein, right? So that's what I have on protein structure, right? So now, any questions so far? Any question? Any question? Any question? Do we have any questions from anyone? Anything that is Umar, are you do you not find answer to the ribbon structure that you see the other time?
I pretty much learned this in human anatomies last semester, and even yeah, pretty much last semester. So everything is pretty like forthcoming to me. Like I understand it.
Okay, that's great. What about every other person? What about others? Any other person? Any other person? Any other person? So let me I'm going to share this screen right now. Let me try to use let me try to use this. Explore in 3D. Explore in 3D. Explore in 3D. Explore in 3D. So just give me 1 second. Explore in 3D. So I'm trying to share something now. Well, so let me try to share this. Okay. All right. So can you see my structure? Can you see my screen? Can you see my screen, please?
Yeah.
Okay. Okay. So this, can you see? So this is a complex protein, right? It's a very complex protein. Because it's a complex protein, that means there are more interaction and attraction. That is why they are coiling. Can you see? And if you can see a lot of helix, alpha helix, right? You can see a form of a kind of simple primary structure around this place, right? And if you put this, it's going to be one amino acid. This is proline, right? I think I show you. I'm just trying to show you this to be able to understand. So there are a lot of interactions among them. They are relating with each other, communicating with each other. So that's why they were able to form a big complex structure. So all those ribbons that you are seeing, this is a typical This is a typical secondary structure because it's from helix, alpha helix, right? Right. Now, when these are a lot of them comes together, that is a complex structure. Oh, Scott, can you please do me a favor? Can you just look for a SARS-CoV-2 spike protein on your camera? I don't have camera on the laptop I'm using, and I don't. So can you please do that and share it with me, please? Share your screen. Just share your
Okay.
screen. Just go to look for a protein, maybe spike protein, and share it on camera. I should have.
You have a
Let me let me see
PDP
if I can.
code?
Just check and use anyone. Let me quickly see if I can download camera here. So just it's better for us to lay this foundation now. Load UCFS camera. Okay, you can share the screen if you have one.
Yeah, this
Oh,
is
good.
Spike and ACE2.
This is Spike and it's true. Thank you. So make it bigger, please. Yes, thank you. So can you see this? So this is just the structure, right? And if you look, this bigger one is the spike, while the other one is the ACE2. The two of them are interacting with each other. They are interacting with each other. So Scott, can you click on one amino acid and see which which amino acid is interacting with that particular amino acid? So if Dr. Scott click on one amino acid now, I don't know if you can do that on camera X and ask it to show you which interaction is that amino acid having with other amino acid. You will see that the calling that you are seeing is as a result of that amino acid interacting with other amino acids around it.
Yeah. It's
I
not
think
letting me.
it's not letting you. Oh, so okay. I'm going to do that. I will make sure I install Camel on this laptop before our next meeting. So, any questions so far? Any question? So we have been able to clearly state protein structure, right? We have, and I've shown it to you. I encourage you to know more of the amino acids, right? Yeah. Can you make it have a kind of okay, I think it's fine. Okay, it's fine. Yeah. So that is, yeah. Can you please show them the sequence? Just show them the sequence for them to be able to know that this is made up of one of amino acid. Yeah, thank you. Can you see the sequence? Do you see all these S that you are seeing? So this is what you have here, is what you have here. So the S is standing for so that's the one letter code for the amino acids. So there is order. If you look at this one, this chain ABC, the first one is methionine, phenylalanine, valine, F. F is phenylalanine, L lysine, V valine, lysine. P is proline, lysine, V is valine, S is serine. I think Q should be tryptophan. I'm not sure. C is cysteine, right? That's how they are. So that's how they are. I mean, now how many of you have heard of variation in SARS-CoV-2 before? SARS-CoV-2 variants. How many of you have heard that before? I'm talking to the students, please. SARS-CoV-2 variants. How many of you have heard it before? Nobody has heard of SARS-CoV-2 variant before? Can you guys hear me? Hello? Hello? Have you guys
I
heard
can
of
hear you.
have
Can
you
you
guys
repeat
heard
the
of
question?
have you guys heard of SARS-CoV-2 variant before?
No.
Oh, you haven't heard of Omicron variant, Delta variant
Oh,
of
yeah.
SARS-CoV-2? Yeah. Do you know why they have variants? Why they have variant is because the original SARS-CoV-2 has a specific amino acid sequence. So when there is a new variant that maybe is supposed to be varying at number 200, right? And another variant, another SARS-CoV-2 now has, instead of varying on that spot, on that 200 position, it now has phenylalanine, right? That is a different variant. So when there is a change in amino acid sequence of SARS-CoV-2, that is when they will say, oh, there's a new variant now, or Omicron, Delta variant, Kappa variant, Alpha variant, whatever variant that they have. Sorry for that. I'm in class. So that's when you have different variants, right? That's when you have different variants. So the sequence is not supposed to be affected, but when there is a change in sequence of the amino acid at a particular position, it can lead to a medical condition or can lead to different variants. Any question? Is that clear now? So now we understand the structure of protein, and we made us know that interactions between one organism to another organism usually happens between proteins and proteins. As we said, for instance, SARS-CoV-2 spike protein interact with human is 2 protein for entry, right? Great. So in our next class, Dr. Scott is going to start by introducing us to the command language that we need for what is called MD simulation. Now, what we'll be doing now is we will teach you how you can. So what you want to do in this way is we want to mimic the interaction that happens in living organism using computer system, right? So to do that, we are going to use different programming language, basically Linux command, right? So starting from our next meeting, Dr. Scott is going to start introducing you guys to Linux command, right? And we are going to probably give you some kind of installation. I think we should teach you how to install Chimera on your laptops right now, right? So then Dr. Scott will take you through Linux command, the basic language you need to learn from Linux, right? And stuff like that. Do you get the point? Okay. So Dr. Scott, do you have anything for them to prepare for against next week, against our next class?
Yeah, we're still working on getting access to the VPN for the students. So we'll have to do, you'll have to work on your laptop for the time being. I can email you some instructions on if if you're on a Mac or a Windows machine, what you will need. Does everybody use Mac or PC? Does it vary? Because Mac has a built-in terminal. Okay, so that's Mac. PC. Okay. Yeah, I can just like email everyone just based on what they have, PC or Mac.
Okay. So I think also by next week, I will show you how to download Chimera. I think there's Chimera for Mac too. So for those that are using Chimera Mac, there is a distribution for Mac, there's another distribution for Windows, right? So I think on the issue of the VPN, I think for now, they can connect to the server when they're on campus, right?
Yeah.
But when they are at home, we can ask them to install mobile extern.
Yeah. If you're Windows, yeah, mobile extern.
Yeah, mobile extern for Windows. Then I think Mac comes with a terminal.
Right. Yeah.
So there's no for them to install mobile extern on Mac. It comes with a terminal already, so they can those people using Mac can do their Linux script and the rest. Yeah.
Yeah.
That's fine. So by next class, we will delve deeply into command and our programming language before we now start to go into molecular docking, protein docking, and evaluating interactions between protein structures. Any questions for me or for Dr. Scott? So
I'm trying to. Well, I'm getting my screen fixed, but it's probably going to be best if I just get a new computer. You said that you guys are still working on the iPads, right? So I
That was
shouldn't buy another
but
one
you
if
but you need your laptop for the work you want to do. Even
yeah,
if
of
you use the
course.
iPad, yeah. So, but the iPad, you can use the iPad to join. We're going to provide the iPad to you. That's not the problem. But you still have to get your laptop to be able to do the commanding, know the work, your project, and to be able to learn this commanding script and programming language that Dr. Scott is going to teach, and what I'm going to teach as well. Is that clear?
Yes, sir.
Okay. All right. So that's one. Number two, my other announcement is there is a Jonas Club. Yes, there's a Jonas Club by our sponsor from Pacific Northwest National Lab in Seattle. So I think I'm going to ask them to send you the link. If you are not in class during that period, I encourage you to please join. Yes, because there will be quite a number of information on things that can benefit you in terms of scholarship, in terms of training over there that can benefit you. Please join so that you can learn as well, right? So I will ask them to add your email address. Oh, Dr. Scott, are you able to upload the student name and email on Confluence?
On Confluence, yes, yeah.
Oh, thank you very
Yeah,
much.
I upload that.
So I think I'm going to share this video also on Confluence so that they can see. They want to see that we are engaging with you students, and they are really happy that at least we started with you. So okay, that's fine. They are paying for you guys, so we need to be accountable to them that we are not just paying good students. You are rich students, right? And they want to see that. All right. So next week, get your laptop ready for next week. Dr. Scott, we start. Then I too will also going to take you some stuff, right? All right. So if there is no question, I think we can call it a day for today. Then our next meeting, let's get prepared for it. If there's any other information during the week, I will let you know. Please, Umar, is Umar still on the call? So please, I think I'm going to, Deja, please, I want you to take note of, if anytime Umar is not on the call, please take note of the attendance. Deja, are you? Yeah. So maybe at the end of it, you can send it on the group that, okay, for this particular date, and I want you to keep that record. We need that record for, even though we are paying you for the first tranche of your payment, if your commitment to the class is not encouraging, I'm not going to recommend your payment for the next tranche, right? All right. Okay, thank you very much, guys. Have a great weekend.
Have a good weekend.
Bye-bye.